Good afternoon, classmates. Good afternoon, Ma'am Dimas. I'm Miguel Nismal, and today we will be presenting our eighth experiment titled DNA extraction. Well, I guess you guys already know kung anong gagawin sa title pa lamang. So, without further ado, let's get right into it. Okay, so right before we start, let me first introduce to you my group members who conducted, examined, and studied the results of the experiment in order to show you guys everything that happened in the experiment. Starting with Miss Catherine Joy Banta, Mr. Gabunyan Gerald Bantawan, Miss Jasmine Marie Cruzada, Miss Mary Angela De Paz, Mr. Raul Miguel Nisman, oh yeah, okay na. And lastly, our leader, Miss Janela Cassandra Vargas. Okay, so let's talk about the theory. But first, let us define what DNA is. So, deoxyribonucleic acid is a chemical found in the nucleus of the cells that contains the blueprint for development and functioning of living organisms. As compared to a set of blueprints, since it contains instructions of how to build cells. The instructions are divided into segments along a strand of DNA and are called genes. Genes provide the code for the production of a protein and control hereditary characteristics such as eye color or personality behaviors. Proteins determine cell type and function, so a cell knows whether it is a skin cell, a blood cell, a bone cell, etc., and how to perform its duties. So basically, DNA is present in every living thing. However, it can't usually be seen by the human eye in its natural state. So by conducting this experiment, we hope that it will isolate the DNA in order to be seen by us. So for the further study, try experimenting with different types of liquid soap. Try switching with plain water to see how the salt affects the experiment, and see if using lower concentration or room temperature alcohol changes your results. Okay, so let's talk about the objectives. So what are the objectives of the experiment? This experiment will seek to answer the following questions. Number one, what is the purpose of the soap in the experiment? Number two, what is the purpose of using ice cold alcohol? And number three, where can the error be in getting only a few threads of DNA in the experiment?
So, these the materials that we will use in the experimentate. Beaker, scoop, safety goggles, salt, hand soap or liquid dish soap, small glass vial, paper cup, alcohol, test tube with tight fitting lid, pipette or dropper, gloves, distilled or bottled water, stirring rod or wooden skewer. So here's the procedure. Create a saline solution in a beaker by adding 2 lab scoops of salt to approximately 25 ml of distilled water. Stir until the salt is completely dissolved. Pour the water into the paper cup without swallowing. Drink a mouthful of the solution from the paper cup and swish it back and forth for at least 30 seconds. Occasionally, scraping your teeth along the inside of your cheeks as you do. It's best to do this with a clean mouth, not right after lunch. So, salt removes protein in the DNA and helps it dissolve in water. Also, it makes the DNA non-soluble in water. So, next is spit your mouthwash solution back into the cup. Then bend the cup into a sort of spout and pour the mouthwash solution into the test tube until it fills about 1/2 inch of the bottom of the test tube. Carefully, add 2 drops of the liquid soap. Membranes are made up of phospholipids, which have similar structure from fats, and the best way to break fats is with soap. Tilting the test tube approximately 45 degrees, use the pipette or dropper on the alcohol bottle to add 20 drops of chilled alcohol so it slides down the test tube without disturbing the solution. Since it's less dense, the alcohol will sit atop the mouthwash and soap solution. DNA precipitates when in the presence of alcohol, which means it doesn't dissolve in alcohol. Next is tightly put the cap on the test tube and very slowly and gently tilt it upside down, then right side up 3 times. Do it carefully so as not to make bubbles. Let the test tube sit undisturbed in an upright position for 1 minute. At this point, you should begin to see a milky white thread, possibly interspersed with bubbles, appear between the solution and the alcohol. That's your DNA. After several minutes, the DNA should be suspended in the alcohol layer. That's it.
So for the first guide question, the question is what is the purpose of soap in the experiment? So to get the DNA out of the cell, we want to tear them open or we want to broken down that DNA. So you presumably seen television commercials that show how dishwashing soap slices through oil. So dishwashing soap contains sodium lauryl sulfate, which disposes of lipids and proteins. Since the layer that encase the cell are comprised of balanced and protein, the dishwashing soap will likewise slice through them. The dishwashing soap isolates the layers and permitting the DNA to be delivered. You can see the DNA you can see the DNA molecule yet because they're dissolved in oil and water, which means that each DNA molecule is surrounded by water molecules. Water is a highly polar chemical. The shared electrons that form the covalent connection between the hydrogen and oxygen atoms are not distributed evenly. This leads electron to cluster closer to the oxygen atom, giving the hydrogen atoms a partial positive charge and oxygen atoms a partial negative charge. The partially positive charge of hydrogen atoms in water molecule attracts the negatively charged sugar phosphate backbone of the DNA.
The second question in the objectives to be answered after the experiment is what is the purpose of using ice cold alcohol? So due to the fact that DNA is soluble in water, the DNA will not be easily visible in only a simple mixture of water, salt, and soap, or in short, in an aqueous solution. When there's alcohol present though, DNA becomes insoluble. As a result, alcohol is required to extract the DNA from the aqueous solution because DNA precipitates when it was exposed to alcohol. However, room temperature alcohol is not permitted to use in this experiment. If performing the DNA extraction, alcohol with 70% solution or higher must be cooled or refrigerated first for several hours. It is important to use cold alcohol because the colder the alcohol, the greater the amount of DNA that is precipitated or solidify and appear and be extracted.
Okay, for the last question of the for the last guide questions, we have where can the error be in getting only a few threads of DNA in the experiment? So first, before going on in answering this question, let us first know what is going through with our experiment. Tiba, we use the salt water as our mouthwash, and you should know by now that some of our buccal cells become suspended in the salt water when we gargle it and spit it back out. So it is said that the more chick cells will gather in the salt water, the more strongly we gargle. And then our chick cells membrane will be broken down upon the addition of this washing liquid. So our DNA gets discharged in the salt water because of it. Well, we all know that DNA is insoluble in alcohol. It solidifies when the alcohol and salt waters collide. As a result, thousands of our DNA molecules are clumped together in the white strings that we see, and it is important to note that single DNA molecules are far too tiny to perceive by our naked eye. Furthermore, when we gargle with salt water, we also gather bacterial cells as well that resides inside our mouth. So the clumped DNA that we see earlier is a mix of our DNA and bacterial DNA. Okay, that's basically what DNA extraction all about. And going back on the question, when we were investigating, we observed that some of us were unable to see clumps of our DNA clearly, but fortunately, Mr. Nismal was able to extract part of his DNA. As a result, after the experiment, we noted some potential errors in getting a few threads of our DNA. First is that there aren't enough chick cells in suspension because the experimenters only risked for 30 seconds, while in other experiments, mouth must be vigorously rinsed for at least 60 seconds to harvest loose chick cells. Well, according to Robbie, he tried the experiment again and vigorously gargled the salt water for at exactly 1 minute and 30 seconds to ensure that some of his chick cells would be extracted. And also, as I've said earlier, uh, dapat strong talaga yung pag-gargle natin. So, more chick cells, will gather or will be extracted in the salt water. Okay, that's for our first potential error. Second potential error is DNA extraction during the experiment must have been poor, or there is a poor DNA extraction. This can be explained by the fact that the samples, like salt water, dishwashing liquid, and water, were not mixed well enough during the extraction, or and the experimenter oversaw the samples. Maybe we can say that some salts were not fully dissolved bago pa uh, tapos ginargled na natin to, or the dishwashing liquid were not mixed well with water. With water, I mean. Okay, third potential error or third factor is the equipment used during DNA extraction. With pipettes, these are the most common tool in DNA extraction. However, in this experiment, the experimenter, just utilized a plastic spoon to extract, transport, or dispense liquid samples. Well, for the fourth one and for the last one, is stated in number two, as or what have discussed by Miss Vargas. It is critical to use cold alcohol because the colder the alcohol, the more DNA will precipitate. Uh, solidify yung appearance niya and be extracted. However, some experimenter noticed that their alcohol began to return to room temperature, implying that the DNA had denatured or broken down. But nonetheless, DNA analysis has numerous applications. It can be used to match crime scene samples, screen for genetic illnesses, or discover new species, for example. Well, it is vital to remember that the extraction of DNA from the sample to be investigated is the first step and the most important step in DNA analysis. Well, basically, that's our only on our observation. And for the observations and results, we have Ms. Depes.
Now for our observation and results, after conducting the experimenters yielded different results. Some of them did not extract the DNA on the first try. Errors for the extraction of the Errors for extraction of the DNA were previously discussed. The experimenters supposedly failed to extract their DNA as there weren't enough suspended chick cell in the solution. The procedures weren't followed enough, such as the salt in the water wasn't dissolved properly and the dishwashing liquid wasn't fully stirred. The experimenters also used alternative materials such as plastic cups and spoons rather than pipette and test tube. This could affect the extraction and the transport of the DNA. Lastly, due to technical difficulties, the alcohols of the experimenters have gone to room temperature. For the results of the experiment, the successful extracted DNA was of experimenter Roby. His DNA was clumped and stranded. It looked like white, thin noodles or a delicate network of limp fiber. The experimenter Janela extracted her DNA, however, the DNA strands were broken. For Catherine and Cabunyan, they managed to extract the DNA but were only in small amount. The other experimenters, Angela and Jasmina, failed to succeed extracting their DNA on the first try but managed to extract theirs after repeating the procedures meticulously. However, they extracted only a small amount. The materials and the procedures must be done properly in order to successfully extract DNA, as this affects the extraction process. The salt in the water must be dissolved properly and must be used to rinse the mouth more or not less than 30 seconds vigorously to collect more chick cells. The dishwashing liquid must also be stirred and dissolved, as this causes the cell to open or lyse, so that the DNA will be extracted in the solution. The alcohol used must also be 70% solution and must be chilled. It is better to chill it overnight before the experiment, as this helps in increasing the yield of the DNA. The cold alcohol protects the DNA by slowing down the enzymes that can break it apart. It can also precipitate the DNA so that it appears quickly and solid. DNA extraction was used for studying the genetic causes of diseases and for developing diagnostics and drugs. It is also important for forensic science, sequencing genomes, detecting bacteria and viruses, and for determining paternity. DNA, once extracted, can also be used for molecular analysis such as polymerase chain reaction, electrophoresis, sequencing, fingerprinting, and cloning.
And that ends our discussion for experiment 8 and or for the DNA extraction. I hope wait lang, ulit nga. Nalimutan ko. Rob, sorry. And that ends our discussion for experiment 8 or for the DNA extraction. Again, this is group 3. Thank you and God bless. Have a nice day.
Tapos kami sinisisi mong nawawala, ikaw yung laging nawawala.
Ito kasi si Robby, Italiano Gold ng Italiano Gold eh.